Well, I'm going to get the ball rolling here. It's 12:01. It is my great pleasure to introduce Dr. Kim Williamson from Uniformed Services University of Health Sciences. This seminar has been in the works for an unusually long time, over a year and a half. In fact, Dr. Williamson was slated to come back in spring 2020 and was going to come the week that the VTLS seminar series was canceled. So it is an extra pleasure to have her here today. So Dr. Williamson is a professor at USU from 2015. Before that, she got her academic start, at least at the faculty level at Loyola University, where she climbed the ranks from assistant to full professor. A couple of things to note about her time at Loyola. She won a couple of awards, one for distinguished university research professor and another for undergraduate research mentoring. And one of the things that impresses me about Dr. Williams' academic record is how much undergraduate research has contributed to her overall output. Dr. Williamson has had a longstanding interest in understanding and interrupting transmission of the parasite that causes malaria. Specifically, she's been interested in the gametocyte stage, which is a sexually differentiated form that can be transmitted from human to mosquito. And she's been interested in this long before it was as trendy as it is today. This, in my view, is both a bold and brave choice, as the gametocyte is very difficult to handle, in my opinion. It takes very gentle handling, and in the case of Plasmodium falciparum, takes 12 days to develop to maturity, and is exponentially more challenging to work with than the asexual stage that most of us malaria researchers choose to focus on. Let me say that gametocyte commitment is a fascinating process that's poorly understood. What specifically triggers gametocyte commitment is only partially understood. Dr. Williams has made some seminal contributions to our understanding of that process, identifying some important genes that promote gametocyte differentiation. More recently, she's been doing some interesting work with clinical samples, looking at host factors that are causally related to gametocyte commitment. And this work has been recently published in a very nice Nature Communications article last year. So with that, I'd like to once again welcome Dr. Williamson to Virginia Tech.
Thank you very much. And sorry I couldn't be there in person, but yeah, pandemic's not quite over yet at this point. So yeah, we'll go with our expertise in virtual seminars. So good afternoon, everyone, and thanks for attending. So today I'm going to be talking just very brief overview of kind of the magnitude of malaria, which probably a lot of you already have a sense of, but just a quick sort of overview as to why we're interested in this, and then a little bit on malaria transmission, and then I'll get much more into gametocyte production, both in vitro and in vivo, and at the end talk a little bit about our some recent work on transmission blocking drug development. So this is just the sort of malaria. This is the death rate of malaria by age over the last 3 decades. So we're starting in 1990 here. And for the first 10 years that I was, actually it also is when I started working on malaria. And so for the first 10 years, every time I was writing an introduction, the number of deaths to malaria were going up. It is primarily in under 5s. You can see the rest of the ages are all down here and pretty flat. Yeah, this is the lion's share of the mortality from malaria. And this is primarily due to the spread of chloroquine-resistant parasites through Africa. This was a great drug. Nobody wanted to get rid of it. It was fantastic, didn't have very many side effects, was relatively inexpensive, and was just, very available. But chloroquine parasites became resistant and just really spread through this population. The other problem was that there was no alternative. So the WHO just didn't have anything else to suggest. Finally, in 2004, the WHO allowed the use of artemisinin derivatives, which had been developed in communist China. Actually, the first article for this is actually, the authors are the Committee on Malaria Treatment, not actually individual authors, so true to form. And then also it found that insecticide-treated bed nets were very helpful. And very rapidly, this really decreased the mortality rate quite dramatically. And this led the Bill and Melinda Gates Foundation in 2007 to announce again that we're going to go another go at malaria eradication. This was a little controversial, but one of the things that really brought up was that we really needed to get a better handle on transmission. How is the parasite being transmitted from person to person, and where's that happening? Are there hot spots? All those kind of questions. Where is this infectious reservoir? And the other thing that kind of links to that is that you see here, there's a plateau that's starting, and that has continued actually. So it's really with the efforts that we're taking now and the amount of money that's going into this, we really can't get this down much lower than it's around 400,000 individuals dying of malaria a year at this point. And that's really where it's stayed for the past several years. Okay, so what we'd like to do is, what I'm going to talk about now is a little bit focusing on this transmission theme. So for malaria transmission, the parasite has to differentiate into a female and a male gametocyte, and these have to be taken up in the 1 to 2 microliter blood meal that the mosquito takes. And then once they get into the mosquito midgut, they're actually triggered to undergo gametogenesis so that they emerge from the red blood cell. This is the female, and the male actually releases 8 little micronemes, like sperm-like guys, and they then fertilize the female, becomes a zygote, continues its differentiation into a kinase, and then that insists out here outside the midgut and forms these new sporozoite forms that then can start a new infection. Okay, so most of this transmission is happening external to the human, which makes it difficult for kind of drug development. And long ago, people started thinking that we could, but what does happen is when you take a blood meal, you actually take up antibodies. So one early approach was to try to look at and they knew that from actually chicken and also mouse studies that you could immunize with these gametes and then get nice transmission blocking activity here. And so they made monoclonals that had this effect. And that's how we found some of the really early surface markers that are now being seen as vaccine candidates. So they're actually some of these PFS 230, which one of the ones I first cloned is actually undergoing clinical trials now. So this is a work in progress. Hopefully, if we can get these working, that will be just such an advance. But anyway, vaccines for malaria are not easy, but there are some human monoclonal. There is a human monoclonal that they developed, and barring the expense of that is another way to go here. Okay, so as people are trying to work on these vaccines, the other option is really drug discovery, and that's again over the past 10 to 15 years has really come alive, and people have been looking a lot more at drugs that actually inhibit these, the gametocytes. This is still a work in progress. So far, only the 8-aminoquinolines actually block transmission well from the drugs that we use commercially or commonly. Even artemisinin and chloroquine, they don't really do much for these mature gametocytes. So they don't block transmission unless you add in primaquine. Talk a little bit more about the dynamics of that later. And so we've gotten a little bit involved in this. At the very end of the seminar, I'll talk a little bit about our work with NCATs at NIH, developing some drug candidates. Okay, so but for the first part of this, I'm really going to talk about the gametocytogenesis. So how do we get these gametocytes formed? And this happens yeah, and this happens from the asexual cycle. So I kind of left this here. We get the sporozoites, they go into the liver. And once they're in the bloodstream, when they pass through the liver, they invade. And then in about 6 days, they've replicated their multiple times to produce 10 to 30,000 new merozoites. And so this form of the parasite, completely different, doesn't look anything like the sporozoite. The protein expression pattern is not similar. So any immune response that was generated here is completely, these guys just completely escape it. They invade red blood cells, and then they have a 2-hour cycle now, 2-hour cycle that they produce. They replicate like 4 to 5 times, and like 16 to 32 new parasites get released, and this cycle continues. The success rate for those parasites is between 4 to 10 new parasites are thought to be generated by each bursting schizon. So this can rapidly escalate your parasitemia, and this is what's causing a lot of your symptoms. But you'll never, these stages, unless you actually took a blood transfusion, took blood and actually transferred it to your vessel, these stages can't get transmitted, so not through a cut or sexual activity. They have to differentiate into gametocytes. And it appears that this, from the data that we have so far, it appears that this sort of, well, we'll talk about it more, but the commitment happens during this asexual cycle. And it appears that during the asexual cycle, this is, you get a few that come off for gametocytes probably every asexual cycle is what's looking for. And it becomes into these, then differentiate into these mature gametocytes. But that takes, as Mike said, 10 to 12 days. And so the questions then for the field are, what levels are people making? What are factors that influence this? How many of these merozoites are actually going to go forward and make gametocytes? All these questions we're really trying to sort of ferret out at this point. And one of the difficult problems that we have, especially looking at this in vitro, is that this 10 to 12 day development period, these parasites are all sequestered. So they're not actually circulating, you can't see them. So the only thing you see when you look in, especially for Plasmodium falciparum, which is the most virulent, you see these ring stage parasites that are circulating, and then you see the stage 5s. The rest of these are all sequestered, these mature asexual and the immature sexual stage parasites. So it's been very complicated. And so just to kind of highlight that, if you are looking just at clinic patients that have come in with, they have symptoms and they screen them. So they found in this study about 1,500 Plasmodium infected individuals by just blood smear. And then of those, only about 10% had gametocytes. And so you can see that this is you've screened a ton of people here, but you have very few gametocytes. So it makes it very hard to kind of really do a lot of this work in the field because it's just difficult to find people that have parasites. And the way you have to do it is to actually get these samples and look at them by RNA to see if they have gametocytes. So it's not a, at site analysis at this point. And very low levels. Remember, I said only 2 gametocytes per microliter are needed. And this is a very low gametocytemia because you figure you have about 5 million red blood cells there in your blood. Okay. And so from doing these sort of molecular detection of the parasite and also this gametocyte, we found that the gametocytes just in the field is found. The gametocytes, about 10%, you're going to be at levels that are actually parasitemic, but you can have many, many more these submicroscopic gametocytes. And yet still there's, why only 10% can we see? Are these the only people that are making them? Is it, we just don't know. So why this variation, the trigger, and then also, is there immunity developed against these gametocytes? This has been another very hard thing to figure out because we don't know who starts to make gametocytes. We just know who has gametocytes at the end. So it could be that, 20% of these people are really immune, and that might be a good drug, good vaccine candidate. So in order to look at this a little bit more carefully with we actually just went in vitro for a while. And so that's where people have been working to begin to get some more strategies to be able to move back to in vivo. So I'm going to talk about our in vitro work and then how that led to some of our work in vivo. So this is just what I was talking about before, is that some of these ring stage parasites, so the merozoite invaded this ring, it starts to develop into a nice schizont with all its new little progeny in there. And this whole process from here to here takes about 48 hours. And then they release and the cycle starts again. But at some population, we have some of these during this ring to schizont population, they get sexually committed. And then when they burst, these merozoites invade a new red blood cell, but they start to develop into gametocytes. And again, this takes about 10 to 12 days for this to happen. And people had known that there were some schizonts that from the 90s from Bruce et al. And Vandenberg's work, two separate investigators, that there's some schizonts had a preference to go this way and some schizonts had a preference to go this way. But the molecular part of that was really unclear as to what's really happening in this. So in my lab, in the course of doing another experiment and cloning parasites after transfection, we found and isolated a gametocyte deficient parasite line. And we also had a parasite line that made good gametocytes. And they theoretically, they were from the same clone, the same population that the same NF54 sort of standard strain. And so we tried just taking the DNA from both of them and probing them, a microarray of all the genes in the Plasmodium. And this is you can see a nice here that most of the genes have a similar signal between the gametocyte positive here and the gametocyte negative. And there's actually only one probe that actually had differential staining, and that was less in the gametocyte deficient. And that probe for this open reading frame right here, and and there were probes around for both of these open reading frames on either side, actually this one and this one, and we knew that they were present in both. And so we just looked sort of by PCR, figured out what this deletion was. It's about a 18 kb deletion. It's actually, this is the largest span that has no other coding region there in all of falciparum. So it was pretty much a gift on this side. And then we have this single open reading frame that was there that we named GDV1 for gametocyte development 1 protein. And it had some nuclear localization signals here, which were interesting, but the Plasmodium has lots of lysines because it's AA, and so these are not surprising actually to get nuclear localization signals, but there was a hint that it might be in the nucleus. And then some predicted alpha helices, but even today, the structure, we don't have any good homologs in other systems. In some of the other Plasmodiums, they're there, but not actually in other organisms. So we don't really have a good handle on what the structural, what the functional possibilities of this is. So we have to, that's our job to try to figure that out. So we do know that it's very critical for gametocyte production. So this is that line that was deficient with the triangles here over these days of gametocyte production. And this is just gametocytemia. And then you can see that we set our cultures up and then kind of just let them go. And they get to high asexuals, and then these gametocytes appear from those high asexuals. So the wild type are here in the squares. And when we complemented this with the open reading frame of and its own promoter, we restored gametocyte production. So that was great. We went on to be able to control this a little bit more. So we tagged it with a degradation domain. So a GFP and then a degradation domain that can be protected by using SHIELD. So when SHIELD is present and binding to the degradation domain, the protein is intact. And when you remove SHIELD, then this degradation domain is obvious, and then it gets targeted to the proteasome and it gets destroyed. And so that allowed us then to have really nice control over gametocyte production. So now we can, so this is in the presence of that SHIELD And in the absence, so in the presence of shield, we get nice GFP expression. And we found that it has this sort of punctate marker. It's hard to see the DNA on this slide, but anyway, these dots are like right around, so they're perinuclear expression right around the nucleus, which is really interesting. We're still looking at that a little bit more. And again, if we had, this is our wild type gametocyte population, if we had shield on either one of two independent clones, we got gametocytes, and then if we didn't have shield present, they're not there. So that was encouraging to like confirm our original hypothesis. And then we could also look at the time course of this expression, and we found that it was expressed from like 36 hours up to about 44, and this is coincident with doing the DNA replication and making that Schizont. So it seems like it's perinuclear as the DNA is replicating, and then you're also probably, you know, transferring epigenetic changes and stuff to this. So anyway, so that's kind of our first two hints at what it might be doing. So it's definitely involved. So in the midst of doing that experiment, so now we know that if GDV1 is present, we get some of these sexually committed Schizonts and produced. And if GDV1 is not there, they all go through this pathway, or primarily go through this pathway. Now, in the midst of while we were working on that work, then Bjorn Kafsak in Manuel Linas's lab, using kind of a similar approach, they also had isolated gametocyte deficient and gametocyte positive lines, and they found that a transcription factor, AP2G, was deficient in the deficient line, and complemented, did all those nice experiments where actually not complementation directly, because this is a huge gene, but did a number of experiments demonstrating tagging it with this shield thing, that it indeed, this transcription factor was required for this. And then another group, Nick Brancucci in Tilvas's lab, was working with heterochromatin protein 1, which binds to histone 3 lysine 9 trimethylated lysine 9 histone 3, which is thought to be kind of an inhibitory mark. And they were looking at the expression profile primarily of some other genes that were known to be suppressed by epigenetic silencing. So they knocked out this gene to see what effect it would have. And so very surprisingly, at least to me, and I think most of the other field, 50% of the progeny, when they did that, became gametocytes, which is quite dramatic. And they were using the shield method. So they had with and without shield. And so 50% became gametocytes. So that's led to kind of a current paradigm where we know at least how a Schizont does not go forward to make gametocytes, or merozoites that make gametocytes. So here we have just our nucleus and, you know, the chromatin and our histones with the DNA wrapped around here. And as many of these can be modified by either methylation or acetylation. Methylation, well, this trimethylation on K9 on these histone tails is thought to allow compaction of the chromatin, and this is protected if the HP1 binds to this. You can't remove the trimethylation, and this is very tight then, and you in loci that have this, you don't get gene transcription here. And so the AP2G lows, they found these marks upstream from AP2G, and in fact you don't get expression. So this is what's happening in asexual replication. And then if you remove this by tagging it with that degradation domain, then you allow the expression of this AP2G. And in fact, that removes it everywhere. So you get high expression levels of AP2G and exposes the promoter for that. And then surprisingly, AP2G actually actually sits down and regulates, it's auto-regulatory. So once you get a little bit of AP2G expressed, then you get more because it will increase. So you get AP2G, but also a number of other genes that are gametocyte specific, and this leads to gametocyte production. And so this has been really nicely worked out by these labs. The question was, where does GDB1 fit into this? What role does it have? And also, this is a very contrived system in the sense that they are, the HP1 is being removed sort of, experimentally. And how this is actually regulated in vitro or in vivo, we don't know how this actually gets removed. So the first question we could ask, again with our shield on and off thing, is GDB1 upstream from the AP2G or not? And so we have our shield on and our shield off conditions, and we're going to harvest then here just at that Schizont period when the GDB1 has been nicely expressed. We'll harvest again at the ring stage, the next ring stage, and then we're also going to see to make sure that they make gametocytes. We treat with NOG a lot because that will inhibit asexual reproduction. So when we set this up, we found that indeed only in the shield on do we get gametocytes, and then the shield off. And this is looking at the fold change between the shield on and shield off. So this is a set of housekeeping genes that aren't influenced at all by GDB1, whether it's there or not, but we get a spike in AP2G, and this is merozoite surface related protein that's also upregulated in these early Schizonts. These are other gametocyte specific genes that aren't upregulated in the Schizont, but when we look at this ring stage, we see this whole set is now upregulated by GDB1. So it appears that GDB1 is acting upstream of AP2G. Till Voss's lab also has had some data that HP1 and GDB1 interact, but a lot more work has to be done on that. That was kind of overexpressed system, and both, HP1 is relatively sticky. So anyway, we have to work on that, but it's possible that GDB1 might be interacting with HP1 and regulating its expression. And so we're working on that in the lab. Those are not easy questions to answer. But one thing that this struck us with this mind towards going in vivo is that indeed, if this commitment really is with these Schizonts, and we can see very nicely that we have these committed ring stages that were produced straight away, that we should be able to go into the field and get these gametocyte committed rings from the peripheral blood and actually see if they're going to go forward to gametocytes. So, and then we can start to look in vivo and see what's going on. So this is how we set, so this is what we were hypothesizing, that circulating in the patient, you're going to have rings and sexually committed rings. You're going to have some gametocytes there also. Okay, so these are what's happening. So we went back, so I was collaborating with Dr. Omar at Noguchi Memorial Institute for Research in Ghana, and we also were, and she was collaborating also with the folks at the University of Cape Coast. And so the plan was to go in and we could harvest peripheral blood. So this would be if we did a blood smear. We have these three different options. You have gametocyte committed, asexual, and then gametocyte ring, or gametocytes in there. And so if we put those into, so the only thing we know about this stage 5 gametocyte was that 10 to 15 days before it was a ring stage parasite. But unfortunately, we don't know anything about that person, right, on that day when this commitment was starting to happen. And so it makes it very difficult to correlate any kind of functional correlates with your commitment. So we put these in a flask, and these guys are going to die out in about 4 days, plus they're very morphologically distinct, no big deal to be able to get rid of them. These asexuals, again, we can get rid of these with this N-acetylglucosamine. And if these were indeed induced, they should go forward and make gametocytes. And we should be able to count these in about, through this time, we should start to see them. And so we can take all kinds of samples and clinical data on day 0. This Festus was the graduate student at Noguchi that was working on this, and put these in culture, take RNA, various things, and then we could just do smears. And so Courage and Lizzie were the folks that were doing all these smears. So we had over 100 patients. There's a lot of for ongoing for 3 years. First year was kind of just a preliminary, but then the second 2 years were our big collection years. And you can see that in ex vivo, these parasites go forward and make very nice gametocytes. Actually, at the same time course, this would be our in vitro parasites. So then very easily we can look at conversion rates here to determine the percent of these rings that made gametocytes by just counting and doing a little straightforward math. And when you do those calculations with all our patients, you could find that there again, as they'd seen in the field with stage 5s, there's a massive, a huge range of gametocyte commitment rates. So some people, a lot of their rings, over 20% of those made gametocytes, whereas about 25% of the individuals didn't make any gametocytes whatsoever. And these were all clinical patients. And about 20% were here in this the high flyer category. So we could use statistical analysis to kind of look at what correlates we had here by looking at multiple variable. And so thankfully here, this is our day 4 and day 8 conversion rates. And those are they are very well correlated with one another, which was good. So our numbers this is just our parasitemias on day 4 and day 8 for the gametocytes. So those are all well correlated, suggesting that they were counting well. And then the other things we found that had could be influence these conversion rate would be the day 0 parasitemia and also temperature. So parasitemia was positively correlated, and an increase in temperature was actually negatively correlated for this population. What we did not find were age, hemoglobin, white blood cell count, did not seem to be correlated at all with our conversion. There were some internal controls here that, age indeed had higher hemoglobins, and we had higher temperatures and the lower red blood cell. So they were age was inversely related to temperature and white blood cell, again, suggesting those younger kids tend to get sicker, which makes sense, which has been seen before. Okay. And so then the other thing that was happening during this, so that was, we have sort of fever and temperature. Matt Marty's group again, actually this is Nick Brancucci, the same Brancucci that was doing the HP1 study, now moved to Matt's lab and was involved in this. They found that they had a parasite line that did not make gametocytes very well unless they removed serum. So they removed the serum, and then because of that, they could fractionate the serum and add fractions back. And so they found that the fraction that if they added back Lysopc, then they would again inhibit gametocyte production. So by having Lysopc around in the serum, it seemed like it was blocking gametocyte production. And so Lysopc, just to remind, I know it's biochemistry, so most of you guys know this, but so this is Lysopc is a derivative of phosphatidylcholine, which is just your glycerol backbone, and then the 2 fatty acids, and then the head group here. And this is the major component of plasma membranes for any mammalian cell kind of thing. And so it can be, the Lysopc is derived by the action of phospholipase A2, and that just removes one of these fatty acid chains. And the phospholipase A2 can be present and get stimulated with apoptosis or inflammation, or actually there's just, this is happening also normally also. So you get Lysopc formed, and obviously it's a little less hydrophilic. And it turned out that in the parasite actually uses Lysopc as its favorite form of this access for choline, and it has to take up choline from the environment in order to make phosphatidylcholine. It has to take up choline if you're going to use choline. There are other ways to get to phosphatidylcholine, but it prefers, this is its preferred pathway. If everything else being equal and it has choline around, it's going to make phosphatidylcholine using the Kennedy pathway going this direction. If Lysopc is limiting, it can also use ethanolamine or serine to kind of come in and do this. And so in the situation where Lysopc is present, your asexual replication is going well. If you limit it, your asexual replication starts to decrease, but your sexual differentiation, your production of gametocytes isn't as affected. So preferentially, you get more gametocytes formed here. And in that line that they had, this is the way that you could actually, see these. It seemed like there was probably actually, this was getting stimulated in their population. It also decreases asexual replication. And so this balance is always there between gametocyte production and asexuals, and it complicates the analysis of these things. But definitely Lysopc kind of was involved in this balance. So we wanted to look at this in our patients because we had it all set up. And so we sent these off just for metabolomics to do like a look at a limited panel of I think it's 148 different metabolites with Duke using their mass spec. And so to be able to try to parse out a difference, we actually decided to look at the high gametocyte producers and the low gametocyte producers. And these are just the clinical characteristics of those two groups. And in fact, we found that the low gametocyte producers did indeed have a statistically significantly higher Lysopc plasma concentration. And the ones that were high, it was lower, whereas these were our controls actually. Neither were different than the control, and we don't know what's driving why this Lysopc level is different at this point. But if we did a receiver operator curve looking at Lysopc and just the gametocyte conversion rate, our prediction is, area under the curve is about 0.78, so about a 78% predictability for this to go, so which is pretty amazing. And the rest of these Lysopc metabolites, not so much. So again, the plans are to look at this in a larger study in the future for this. So in summary, at this point, we have this variation. So parasitemia seems to be associated with increased gametocyte production, fever, and Lysopc with decreased production. And then the rest of these didn't have any effect at this point. And so it suggests that it's possible that maybe at low, when you initially are infected, you have low parasitemia, you have high asexual, you want to start really revving up your asexual replication, and you're not that interested in sexual differentiation at this point because you need to get enough asexual parasites up to get level. So when you do convert, you have enough gametocytes in there to be taken up by a mosquito. So it might be, and this is possibly when you're at low parasitemia, you have high Lysopc levels. Then as your infection gets established, perhaps you're lowering your Lysopc levels. This is unclear at this point as to what's, how these two are linked. We don't have any indication that these two are linked. But it's possible that maybe because the parasite's using more of it, it's at least in a limited area, that you've decreased the local Lysopc levels. And this may be a trigger to augment sexual differentiation. And you're at high enough parasitemia now that if you make some gametocytes, you're going to have enough to be able to transmit to the mosquito. And then if the immune response gets initiated, perhaps through fever, again, this is all speculation, then that's another sign that asexuals are going to be killed off, and that perhaps you need to head back towards asexual replication away from sexual differentiation to maintain levels of the parasites to kind of make it through that population. And so that's our working model for this. Clearly, we have to expand this to larger populations, different regions, and also look at asymptomatic carriers to see if there's a difference, because it seems like asymptomatic carriers have very few parasites, yet they still have a decent number of gametocytes. So looking at that ratio, is it different when you're sick or when you start to have symptoms or not? But in order to do this, not that it didn't work well for our ex vivo, we really need to define these molecular markers to be able to take the blood from someone and actually say, okay, this is what population of your parasites are committed to rings by doing a test in vitro as opposed to doing that ex vivo study, which it was fun, but I don't think anybody wants to go back to. And people are willing, they can try it, but it was a lot of work for all that culturing. That's a lot of culture. Anyway, so we took a similar approach that we did for Lysopc. We divided our individuals into high gametocyte conversion rates and low, and looked at their transcriptome to be able to look again to try and see if we could find some good markers that would be indicators of this, and also to look just what's different about these two populations of parasites. Can we get some hints as to what genes are involved in the function of these things? And so this is the sort of the volcano plot. So we had a number of genes that were, so we have our p-value here and our fold change over here. These are the set that were upregulated here. Yeah, and so this is their gene list, which I'm sure doesn't make too much. Some of you might have some genes that are interesting, but so just to parse it out a little bit, we have, this is our AP2G was really very strongly correlated with the high GCR, which was good because we know that this is, the levels here are, driving a lot of this. We also interestingly found a number of these surface molecules. So this is a family in the parasite that tends to be on the surface of the infected red blood cell, but people haven't really been able to really figure out what the function of these are. But we have a number that are specific for this population. And then a number of just a couple enzymes here. So these are both histone deacetylase, and this is a lysine, demethylase here. And so both of these involved, again, in epigenetic regulation. So we're interested in pursuing these guys. A ROM is this big complex that's involved in I'm going to, oh well, I forget the product of that. Yeah, haven't looked, yeah, sorry about that. And then these are a number, the rest of these all have export sequence. And that kind of fits in with early on that these exported proteins sort of modifying the environment of the red blood cell is one of the first things that's distinct about being a gametocyte versus going forward to asexual replication. Yeah, so we're interested kind of in all these molecules. What was interesting, what wasn't there. So we found the number of genes that were not there. These are some of these early, the ones up here at the top, were these genes that were early on that are gametocyte specific, but those we didn't really see very many in these circulating rings. And that leads us to suggest that maybe these aren't expressed until the parasite's actually sequestered. And that this sort of is a marker that these are a little bit later in development that we would see in vitro, but we wouldn't see in vivo. And then some swing things here that these guys are expressed later, so as well as earlier. So these are kind of more long-lived expression profiles, and these kind of vary a little bit more. These, so we wanted to look at these first guys and see what can we really find as a good marker. And so these are the top three, the APTG, the surfin, and the two different surfins. And in our low population and high population, see nice distinct populations, so that when you have high GCRs, you really don't run into the low GCR population. So you can separate these quite nicely with these three genes. And when this was just a cohort again of high and low, this was an independent cohort. We selected from the individuals. And then here, we just look at all the individuals that we had in the population. And you can see that, again, there's a really nice correlation between the expression of those and gametocyte commitment rate. So we have three nice markers now that we can actually start to use to parse this data out a little bit. And so now we're back off to school to monitor asymptomatic kids and see just kind of what populations they have in their that are circulating. And in a preliminary study of this, and we're also doing it every 2 weeks, so we can kind of see if that correlates them with circulating 5s in these individuals. In a preliminary study, we found that in fact, these kids that are appear completely healthy, about 89% of those at some point during this 10-week period, this is after the malaria season, actually had parasites. And that of the ones that were infected, about 48% had nuclear, at least submicroscopic detection of gametocytes. So again, it points to this huge infectious reservoir and the real importance that there is to be able to deal with this, either a vaccine or drugs that are going to eliminate this, or it's going to be very, very hard to eliminate malaria. Very difficult. It's just, yeah, it's mind-boggling. And about 20% of these guys who had gametocytes during this 10 weeks, they were at levels that would be able to be transmitted, or hypothetically labeled to be transmitted. So it's a real big population, like a huge population. Anyway, so that's going to tie in. So that's kind of where we are with the in vivo. Now I'd like to switch gears a little bit and talk just for a few minutes on our drug development work. I'll try to be quite quick about this so people can, yeah. So this is kind of just at the very beginning of this. What the big problem was, we didn't have anything that we could kill a gametocyte with, which, yeah, was bad. So none of the current drugs really did. Primaquine needs to be metabolized to be able to work. So nothing in vitro would kill gametocytes. They wouldn't be produced, but we couldn't kill them. And so one of the first things we were looking at are these cysteine proteases. And I primarily show this to show how insensitive these stage 3s are. These are 2 that have been treated. When we infect the earlier stages, either asexual parasites or stage 2s, we get this really cool phenotype where you can't digest the hemoglobin. So you get this huge food vacuole that gets expressed. And here in the stage 2s, we have these, instead of coalescing into one major food vacuole, we have little individual food vacuoles here, and you can see all of them swelling up like that. So in looking at these cysteine proteases for another reason, we tried a different set of them, one that was a little bit more, and lo and behold, one day we came in and smeared them and no parasites. They were just, little tiny blobs here. And even when we put them on stage 3s, it would form these really weird structures and sort of needle-like parasites. And we were like, this is amazing. How can it be so different? We thought these were cysteine proteases. But in talking with Matt Boggio, who does a lot of work on both cysteine proteases and proteasome inhibitors, he thought that it might be that the cysteine protease inhibitor that we used had also proteasome activity. And so when we used a specific proteasome inhibitor, we got that same phenotype. This is with epoxamycin. And so epoxamycin is quite good at killing even late stage. This is a stage 5 gametocyte. And so even if you add this to these in this mature population, in 24 hours, you start to see, especially at 100 nanomolar, you're going to start to see some changes. And then even at 72 hours, they just disintegrate, which is really nice, not very useful. Well, actually now they've just started to get some proteasome inhibitors that are more falciparum specific. So I think that's going to be an interesting future area to watch. Very tricky. The proteasome is like so conserved, very difficult. But with this in hand, we did a repurposing screen. So we've developed a screen that we can use in the 1594 well plates, and with NCATs at NIH. Actually, Wei Sun was critical in doing this, and Wei Zhang there. And so we screened like 6,000 compounds, and we only got 68 that actually had that did much to the viability of these late stage gametocytes. And so the top hits are here. These are all EC50s at 50 nanomolar or less. And there's some old friends here, which had already been shown, another proteasome inhibitor, and this was dactomycin, had been previously shown to affect gametocytes. And then ionophores had been known to be very effective in chicken, in avian coccidia, actually. And then Taurin 2 is one that came up here. And so this is an inhibitor of mTOR, the mammalian target of rapamycin complex kinase. And so the chemists at NCATS thought this was intriguing and that this might be more tractable. Maria Moto at the same time had sort of through this time had also come up with seeing that Taurin 2 actually inhibited malaria parasites also. So this is Taurin 2, and you can see, whoops, sorry, let me go back. This is Taurin 2, and this is just its effect on hep G2 cells, so quite ineffective against hep G2 cells. And then here's the gametocyte inhibition profile and the asexual profile. So down here in that nanomolar range for these guys for IC50s. What was also intriguing was that there was Taurin 1 that we found, and this had very little activity on the gametocytes, although it's really pretty similar in structure. And then in order to kind of test to see if these side groups were, which one was most important, we have, they made this compound. It's always nice working with chemists because they just kind of make these things, and found that in fact, this is very effective. So indeed, this 2-aminopyridine is very important for the activity, and this group is really irrelevant. So two things. So that's nice to know that this is kind of the critical active compound, but having an extension out here didn't affect activity at all. So that meant that we could tag this with other things and make some affinity matrices and stuff, which I'm not going to talk about here, but it was kind of nice initial structure activity function. So this just shows that Taurin 1 and then the Taurin 2 and the new compound. And though these two compounds are very similar against mammalian mTOR, so it kind of gave us a sense that maybe the parasite does not have an mTOR related kinase, protein kinase at all. So it just, we might have a Plasmodium, might be able to create something that was Plasmodium specific. And so they made many, many compounds. Actually, this was early, so we were probably in the couple thousand compounds at this point. And one of the most interesting was this 3710 compound, which retained its asexual, not quite as good against gametocytes, but its asexual activity was there. It was much less active against this human mTOR. And so this is just the ratio there between these. And it had relatively good solubility and microsomal stability, better definitely than TOR. And yeah. And so we went forward. We'd already tested this in gametocytes and asexuals, and in liver stages. I'm not going to go through this in the view of time, but it's quite easy to see here that this is our vehicle where so this is IVIS. This is using a mouse that has expressing luciferase, and it's infected with Burgii that's expressing luciferase, and you can just track the mice over time by using by scanning the luminescence here. And so you can see that these guys get very ill, parasites everywhere, and they all died by day 31. Same thing, if we gave the drug prior to giving the infection, they all died. If we gave it on the same day as the infection or after the infection, you can see here that in 24 hours, this is when we applied the drug. There were some parasites, but then they were cleared. And then various concentrations down here. This is pure methylene. And so again, it was very effective now. So this works on asexuals, it works on gametocytes in the blood stages, and it also works on the liver stages. It does not work so well on hypnozoites at this point, but it's very good for Plasmodium falciparum to eliminate the developing stages of the parasite. So that was very nice. Then we wanted to know what does it target? And so to do that, we generated resistant lines, and we had to do this by slowly ramping up the taurin concentrations, taurin 2 concentrations over generations. And we finally got 2 independent lines, B5 and C3, and found that when we sequenced, did no whole genome sequencing, C3 in the lines that were resistant had a mutation in the active site of PI4 kinase, which is not totally surprising because this is a lipid kinase, not a protein kinase, but the structure is similar to the mTOR. They're related to these kinases. And so this is right in the active site. These are some other mutations that people had also found in PI4 to be associated with drug resistance. And we also checked, and in the other line, it didn't have any mutations here, but it was duplicated in that region. You just see, so if we just do quantitative PCR, you can see that this region is highly upregulated. We get a lot more copies. And so this is that duplication region. It's about 4 genes with there. And then, so just to remind people what a PI4 kinase does, this is again, our lipid, so our phosphatidylinositol, and it has the head ring here, and that kinase is going to phosphorylate in the 4 position on the inositol ring. And this primarily goes from PI to PI4, and that can start a whole series, a number of these, especially this guy can be involved in signaling. And this also drives membrane transport and things. And we also checked to make sure to see now, unfortunately to us, it was a little disappointing, a really nice finding, but PI4 has been the target of other inhibitors, and actually a number of classes of inhibitors that are effective against PI4 and kill the parasite at definitely at all these stages and things. And so we wanted to check and see if indeed our resistant lines were resistant to these other PI4 inhibitors, and indeed they were. So this is one that MMV was working on. These are 2 that Dr. Winsler's group developed, and this is the one that we developed with NCATs. And this was also another, so this is DD2, which is a different parasite line, and Dr. Winsler's group had actually added in another copy of PI4 kinase to increase the levels of PI4 kinase in this, and indeed you can see that that confers resistance to our drug as well. So kind of, this is just that same pathway I showed before. This is PI. The PI4K can take you this direction. And then with various other PIP kinases, you can get all these different forms, signaling forms of phosphatidylinositol generated. This is the Plasmodium thing. So we don't have this class 3 kinase that can put, it's not thought that we can have this one that can put the 5 on here. So we have kind of a truncated pathways, but indeed the PI4 these are these 2 other, this is the MMV48, and these are the 2 groups, the KU56 and the the KU56 is this group. And so they are all inhibiting this PI4, and now we can add in that taurin 2 and their analogs do this also. And it appears that from the different phenotypes, this membrane trafficking is very important from Golgi to plasma membrane, and could, affect protein trans export. And also just more recently, the Malcolm McConnellville showed that the taurin 2, there's a decrease in hemoglobin-derived peptides. So it seems like it might be doing something in the food vacuole, maybe due to altered membrane and protein trafficking. Little unclear as to what it's driving there. But my sense is with this, if you're not getting good trafficking from the Golgi to plasma membrane, you're going to be messing up many things, which is why it's so effective at all stages of the parasite. These guys, they were checking in asexuals as opposed to in gametocytes. And we saw before that, decrease in hemoglobin might not be having too much effect here. So I think, this is a very promising candidate from a functional point of view. The real issue is whether you can generate it as a human, get a Plasmodium specific compound. So NCATS is working hard on that to try and see if they can increase the specificity for Plasmodium, which would be great because it works really nicely, even really late into gametocytes, so probably in the midgut. Okay, so that's kind of where our story is at this point. This is just, sort of our basic summary. We've got the GDV1, this critical upstream regulator of AP2G. We're working out how that's actually happening in the parasite, and then starting to, again, we've generated now these really nice biomarkers that hopefully we can go in and really look at, what's happening in the peripheral blood and what factors are involved in gametocyte commitment, but also do the molecules that you see, the proteins you see that are committed, do they actually make it to stage 5 by monitoring do we have stage 5 parasites in like 2 weeks? And so that's what we have, these, more ongoing trials at this point for this. The nice thing about working on this in asymptomatic patients is that you don't have to treat them until they're sick. If they do get sick, we do treat them. And then they stay in the study, but we treat them. And also theoretically, by knowing if the person has gametocyte committed rings, then you could also go in and treat them earlier to make sure with permaquin or something to make sure that they don't continue to develop into mature stage 5s. Many things, and then really trying to work out the PI4K and how, if we can develop some nice Plasmodium specific inhibitors. So with that, I just like to, this is a team of definitely lots of people involved in this work. So I'll stop there.
All right. Let's give Dr. Williamson a virtual round of applause here for a fascinating seminar. If there are any questions, so the way the questions will work is if you post them in the chat, I will moderate and communicate the questions to the group. So let me start off. I was intrigued by the perinuclear localization of GDV1. Have you delved into that at ultrastructural resolution, and specifically, is that a subdomain of the nucleus or is it outside the nucleus? What's your...
Yeah, so from our it appears that it's internal to the nuclear membrane, but right on the edge, actually. And we've attempted to go further with immuno-EM and stuff, but we don't have. What's been a real problem is trying to make GDV1 recombinantly has been difficult. We tried, and we tried to get some antibodies. So we don't have antibodies directly to GDV1 despite multiple attempts on that, which it would be so nice if we had them, but we don't. And so what we've been using is to try to use antibodies against GFP, and it just hasn't worked that well. The thing that I can tell you is that it is like a rock in there. So then we also tried to do pull down experiments with, because we have a GFP tagged, right? So there's lots of nice reagents to be able to do pull downs and things. So it appears that it's locked in there. I don't know how it's associated, but we need to use 8 mole urea and 10% SDS to get it off. And by that point, you've actually destroyed the hemozoan crystals are no longer in the thing either. So it's amazingly attached.
To DNA? Or...
Well, we tried, I don't know what it's attached to. I think it might be some cytoskeletal thing, because we tried, using the DNA disruptor, benzamidine, but that's not the right word. Anyway, so benzoase, I think, is the one that is one of the DNA that degrades DNA. We've used a number of DNases to try and get rid of the DNA. And we've also, So it has not been easy. It always ends up, we can get a little bit off, but not very much until you go to these drastic measures. And then when you do that, you can get it off the population. So I guess
nothing's binding it to it at that point. So
right, that's the problem, is that then you have to do cross-linking. So what we're trying is to do BioID, but we haven't been able to tag it with BioID yet. And I'm hoping that it's been technical issues that, you know, sometimes transfections work and don't work. We just haven't gotten parasites up. So we don't know if the BioID is kind of killing the parasite because it's, you know, there's biotin in the media and maybe it's biotinylating things that shouldn't be biotinylated. And so we haven't gone honestly yet to growing it in biotin deficient media to do our transformations. But what we did try was to put a DD domain on the biotin because we know that, you know, they grow fine if you have GFP, if you have GDV1 GFP degradation domain. So it would be nice if we could just do all the cloning and with GDV1 biotin, the biotin ligase, and then with DD domain. And so everything would be degraded until we put back the shield, and then we have the biotin, we have the molecule. And so that would be a wonderful experiment on paper, but we have to get it in the flask, and it hasn't happened yet. So that's what we're trying to work on with this. It's also anyway.
Yeah. By the way, so there is a Q&A section as well. I'm also looking at that, so you can post your questions in either. So a question about the structure prediction. So this AlphaFold, these AI driven protein conformation prediction algorithms have turned out to be really, really effective. So have you considered using AlphaFold or
I would, yeah, and we have not done that since we have not done that yet. Is there a site for that, or do you have to talk with them directly, or?
I'm not totally
If
sure,
somebody, I'm very open about sending this, you know, the sequence around to people to look at this because, you know, we used iTezer and the Fire are the ones that we've used. We haven't, it would be really cool to try this with the AlphaFold. Yeah,
Fire is not really, you know, Fire looks for structural homology. But anyway, so yeah, I think.
Right. No, but that's what's out there, right? I mean, I don't.
Yeah, David Baker's approach, I think, is really interesting. So anyway, we can talk later if you're interested.
Okay, yeah, no, because I'm very interested in trying to get some structure. I did just a generic structure. It was kind of had all these beta sheets and sort of formed like a semicircle. So it looked like it could sit on something, which was very interesting. But we would love to get the targets. We have so many things that we want with this.
Yeah, okay, so another question. Any idea how so many groups ended up finding inhibitors of PI4K?
Well, I think it's a good target. So I think a lot, I mean, there was a huge amount of work, and there are not a ton of targets, it seems like. There's the ATPase4 that was a number. So it seems like many of these that have these enzymes that are involved, it seems like people have done so many libraries recently. It's just been the thing to screen libraries. And so these sort of, I don't think it's necessarily low-hanging fruit because we don't have any drugs from them directly, but these enzymes that are critical seem like they've gotten tagged. A number of them have been tagged, and when they have, it seems like they have different compounds can all activate them. So the PI4 is definitely a very good, it's needed, it's very essential, I guess.
There's also, it must be inherently druggable.
Well, actually, I shouldn't say that. It's a kinase, right? So people have been looking at kinases for drugs forever. And these are, you know, it's the ATP, most of these go into that ATP site. So I have a feeling that people have been designing a lot of compounds to try to kill off kinases.
Right.
But yeah, so they're all ATP site binding things. So I think that decreases their specificity, unfortunately, for Plasmodium. And it's been a problem. Yeah, unfortunately.
Yeah. So a question on the patient studies. Do patients that get reinfected have a similar gametocyte conversion rate between the two different infection events?
Yes, so we do not know that yet because we're just starting. We have all the RNA in our freezer from our big field trial. And so hopefully I will be able to tell you that in the future. Good question.
All right, well.
Apex, yeah, we're definitely looking into other alternatives to BioID. We were using BioID 2, which is small, but then I think there are some that are even smaller. Yeah, anyway.
Is what a lot of people are using now. All right, so I think that does it for the questions. So seeing as we're past the hour, I think we'll wrap things up. So thanks again for a very interesting seminar, an update on what's been going on, and yeah.
Okay, great. I see somebody added in the link, so that's good. I will talk with them.
All right, I'll talk to you in a little bit then.
Okay, bye-bye.
Bye.